Imagine a spy operating deep behind enemy lines. They're constantly changing their disguise. A new mustache on Tuesday, maybe a completely different jacket on Wednesday, maybe even an altered walking gait by the weekend.
Right, just totally changing how they present to the world.
Exactly. And they do this to avoid capture. But beneath all those elaborate, super convincing costumes, they never actually change their true identity. I mean, they're the exact same person, just wearing a different biological mask.
It's a great analogy.
Welcome to the Deep Dive. We are looking at this absolutely groundbreaking 2026 paper from Cell Host and Microbe. It's titled, Epigenetic Phase Variation in the Gut Microbiome Enhances Bacterial Adaptation.
Yeah, it's quite a mouthful.
It is. But the reason we're diving into this today is that this spy trick, changing the outer disguise while keeping the core identity perfectly intact, that is exactly what the bacteria in your stomach are doing right now to survive massive stress.
Oh, yeah, like surviving a literal nuclear bomb of antibiotics.
Right, and doing it without relying on permanent genetic mutations. Have you ever wondered how your gut microbiome bounces back after a harsh round of antibiotics? Well, the secret is hidden in this paper.
It really completely changes how we view bacterial survival. And, this research by Nye and colleagues is just phenomenal. What makes it definitive is the atom space.
Yeah, tell us about that, because they didn't just guess, right?
No, not at all. They didn't just sequence the gut bacteria using standard methods. Standard methods often just chop DNA into tiny, unreadable fragments. Instead, they built a custom discovery algorithm.
Okay, what does that actually mean for the bacteria?
Well, they used long-read metagenomics. So instead of chopping it up, they pulled out these massive, unbroken chains of DNA to see the entire physical scaffolding of the genome.
Oh, wow. So they could see the whole picture at once.
Exactly. And then they combine that with single-molecule methylation analysis to actually detect the microscopic chemical tags sitting on top of that DNA.
And they did this in live animals, right?
They did. They proved the whole mechanism in living, breathing, in vivo mouse models. So this is the absolute highest tier of molecular detective work. We aren't just looking at a petri dish here.
We're seeing the gears turn in a living system. So, okay, let's get into the mechanics of this. Because to understand how the bacteria survive, we have to understand that disguise. We are talking about DNA methylation.
Right, the core mechanism.
So if we think of a bacterium's DNA as, say, the master blueprint of a house, methylation is kind of like taking a neon highlighter pen and coloring over specific lines of that blueprint.
I love that. And depending on what you highlight, the contractor building the house will read the instructions differently. In bacteria, there are 3 primary chemical tags, these highlighter colors, if you will, that get added to the DNA.
Okay, what are the colors?
We have N6-methyladenine, which is usually just called 6-MNA.
Oh.
Then N4-methylcytosine, or 4-MMC, and 5-methylcytosine, which is 5-MSC.
Got it. 6-MA, 4-MMSC, 5-LNC.
Right. And the physical enzymes that literally walk along the DNA and attach these tags, they're called DNA methyltransferases. Or just MTASes for short.
MTASes, so they are the ones holding the highlighters.
Yes, they are the writers. They scan the master blueprint. They look for a very specific sequence of letters, and boom, they stamp a methyl group onto it.
Okay, so the MTASes are the writers. But the paper introduces this concept called epigenetic phase variation, or EPV.
Yeah, EPV is the star of the show here.
So normal phase variation is when a bacteria literally flips a genetic switch to mutate and change its physical traits. Right?
Exactly. It changes the actual letters of the blueprint.
But this is epigenetic. So the bacteria isn't changing the fundamental letters, it's just changing the highlighter pattern. It's basically generating completely different survival traits from the exact same genetic code.
And that is the crucial difference. It's brilliant. The researchers found that this epigenetic variation, this EPV, happens in 2 distinct ways.
Okay, let's break those down.
The first is genome-wide EPV. This happens when the bacteria undergoes a structural variation. Imagine a physical shuffling of the DNA within the MTAS gene itself, or its targeting subunit.
Okay, a physical shuffle.
Right. And the most common way this happens is through something called an inverted repeat.
Wait, I really want to visualize this. What is an inverted repeat actually doing in physical space?
Okay, think of a small section of the DNA like an old school cassette tape. The bacteria essentially ejects a tiny piece of its own genetic code, flips it 180 degrees upside down, and pops it back into the sequence.
Oh, literally inverting it.
Yes. And when that sequence flips, the MTAS enzyme that gets built from that code fundamentally changes its physical shape. Its targeting system is completely rewired.
So it starts looking for a different word to highlight.
Exactly. So instead of highlighting one specific pattern across the entire bacterial genome, it suddenly starts targeting a completely different pattern. And because it affects how the writer operates everywhere, it alters the gene expression of the entire cell all at once.
I see. So it's like flipping the master breaker in a house. You hit that one switch in the basement, and every single light in every single room changes color or goes dark simultaneously. It's this massive systemic change.
A master breaker that randomly rewires itself, yes. That's genome-wide EPV.
But then the paper details a second type, right? Site-specific EPV.
Right. So if genome-wide is the master breaker, site-specific is much more subtle. It's highly localized. It usually involves just a handful of methylation sites on the DNA, specifically in intergenic regions.
And those are the areas between the genes.
Yes, specifically the promoters. The docking stations that sit right in front of a gene and control whether it gets turned on or off. Site-specific EPV is driven by competitive binding. It's basically a microscopic turf war.
So if genome-wide EPV is the master breaker, site-specific EPV is more like a dimmer switch in the kitchen. You're just adjusting the lighting for one specific room without touching the rest of the house.
It's a dimmer switch, yeah. But imagine you were trying to turn the dial, and someone else's hand grabs yours and physically fights you for it.
Oh, wow. Who is fighting who?
The MTACE, our writer, is trying to add a chemical tag to a specific parking spot on the DNA. But a transcription factor, which is another protein trying to turn the gene on or off, wants that exact same parking spot.
Okay, so it's a race.
Exactly. If the transcription factor beats the MTACE to the spot, the DNA doesn't get methylated there. The local environment, the stress the bacteria is under, really dictates who wins that wrestling match.
Okay, so before we dive into how this actually plays out in a living host, I want to make sure we have this mental model locked in for you, the listener. It's time for our first quick recap. Let's bullet point this.
Sounds good.
Okay, point 1. We have our MTACE writers, and they are stamping chemical tags onto the DNA to change how it's read.
Point 2. Genome-wide EPV. When the bacteria flips a piece of its own code, that inverted repeat, it changes the writer's targeting system.
Which alters the entire genome, like a master breaker.
And point 3. Site-specific EPV. Sometimes it's just a localized turf war at a single gene promoter, like two hands fighting over a dimmer switch.
Perfect. So put yourself in the shoes of that bacteria for a second. If your entire world is suddenly flooded with toxic chemicals, do you pull the master breaker or do you just wrestle for the dimmer switch? Keep that in your mind because we are going to look at a real-world battlefield right now.
Yeah, things get intense here.
Let's look at the first clinical case in the paper. The infant, amoxicillin, and the genome-wide shuffle.
So the researchers analyzed data from the ECAM study. These tracked the gut microbiomes of early childhood infants before and after they were treated with amoxicillin, which, as you know, is a very common, very powerful antibiotic.
A total nuke to the gut.
Right. And the data they pulled using their discovery algorithm was just striking. First, they saw this with a bacteria called Bacteroides zellanosolvens, which had 2 pairs of inverted repeats, creating 4 distinct genotypes.
Okay, 4 different disguises.
Yeah, but then they found the real star of the show, a specific strain of bacteria called Akkermansia muciniphila, and it relies incredibly heavily on this genome-wide master breaker system to survive.
Let's focus on this Akkermansia strain, because its mechanism is just wild. It doesn't just have one inverted repeat. It has 3 nested inverted repeats.
Yeah, a sequence inside a sequence inside a sequence.
It acts like a literal slot machine. When those 3 sections of DNA shuffle and flip independently, they create 8 different structural combinations. And those combinations create 6 completely distinct epigenetic phases, which the researchers labeled M1 through M6.
6 different disguises for our spy.
Wow.
So picture this. Before the infant was given any antibiotics, the environment in the gut was peaceful. In that peaceful environment, the M1 phase was completely dominant. The vast majority of the Akkermansia population was wearing the M1 disguise, meaning their MTACEs were highlighting one very specific motif across the genome.
Already doing the normal thing.
But then the infant is prescribed amoxicillin, the drug hits the gut, and that dominant M1 population is absolutely decimated.
Left off the map. Gone.
Completely. But the Akkermansia as a species doesn't go extinct in that infant. because surviving in the wreckage of that antibiotic bomb are the M2 and M3 epigenetic phases.
Okay, I remember the slot machine.
Exactly. The bacteria in the M2 and M3 phases had a shuffled sequence. Their MTase enzymes had a different physical shape, so they were highlighting a completely different set of genes. Suddenly, while M1 is dying, M2 and M3 multiply and take over the entire gut.
But wait, I have to ask, how does changing the highlighter pattern actually stop an antibiotic from tearing the cell apart?
Yeah.
Like physically, what is happening?
Well, the researchers found the smoking gun. It all comes down to a specific gene called mucC. In the M2 and M3 phases, the new methylation pattern essentially supercharges this mucC gene. It turns it on high.
And what does mucC do?
The physical mechanism of that mucC gene is what creates the survival trait. When mucC is upregulated, it fundamentally alters the bacteria's cell envelope. It changes the membrane permeability and essentially thickens the physical barrier around the cell.
Oh, I see.
Amoxicillin works by attacking the bacterial cell wall. But if the mucC gene is working in overdrive, it builds a fortress wall so thick and resilient that the large amoxicillin molecules simply can't penetrate it effectively.
It literally puts on heavier armor. That's incredible. But to prove this wasn't just a permanent genetic mutation, that the bacteria hadn't just evolved to become a superbug permanently, the researchers introduced a brilliant twist, didn't they?
They did. They took these heavy-armored M2 and M3 bacteria and exposed them to a completely different class of antibiotic, ciprofloxacin.
And how does that one work?
Ciprofloxacin doesn't attack the cell wall at all. It gets inside the cell and attacks the DNA replication machinery. And in this new toxic environment, the heavy mucocell armor is actually a huge metabolic liability.
Because it's heavy.
Exactly. Maintaining that massive fortress wall takes a ton of energy. So under ciprofloxacin stress, the bacteria completely reversed their strategy. The M2 and M3 populations died off, and the original M1 state, the lightweight, vulnerable to amoxicillin state, suddenly thrived again because it wasn't wasting energy on useless armor.
Okay, I have to stop and push back on this, because this implies a level of intelligence that bacteria just do not have. I mean, they don't have brains, they don't know the amoxicillin is coming, and they certainly don't sense the ciprofloxacin and think, oh, time to drop the heavy armor and flip our DNA sequence.
No, of course not.
How are they adapting their disguises so perfectly in real time if they are completely blind to the future?
That is the genius of it. They aren't adapting in real time. They are doing something called epigenetic bet hedging.
Bet hedging, like gambling.
Think of it like a highly diversified financial portfolio. The Akkermansia population doesn't wait for the antibiotic stress to arrive. Even in a perfectly healthy, peaceful gut, where the M1 phase is thriving and multiplying rapidly, the bacteria intentionally force a tiny, tiny fraction of their offspring to flip those inverted repeats.
Oh, wow.
Yeah, they constantly keep maybe 0.1% of their population in the M2, M3, middle 4, middle 5, and middle 6 states.
So they're constantly maintaining all the disguises in the background all the time.
Yes. They willingly sacrifice the metabolic efficiency of a tiny fraction of their population just in case the environment suddenly turns hostile. When the amoxicillin drops, the 99.9% of the population in the M1 state is slaughtered.
But the bacteria don't care.
They don't care because that 0.1% in the M2 and M3 states were already wearing the thick mucC armor. They survive, they multiply, and they rebuild the colony.
It is literal evolutionary gambling, but they are playing the odds perfectly. They never put all their chips on one number. Let's take a breath and synthesize this. Time for our second quick recap.
Bullet point time.
Okay, the Akkermansia bacteria use nested inverted repeats to constantly shuffle their writer enzymes, creating a portfolio of different epigenetic states.
Right, and when amoxicillin hits, the dominant M1 population dies, but the rare M2 and M3 subpopulation survives because their specific methylation pattern forces them to upregulate the mucC gene and build a thicker physical wall.
And crucially, because this is epigenetic, it's completely reversible if the threat changes, like we saw with the ciprofloxacin.
It is a dynamic, constantly shifting defense grid.
So listener, think about your own life for a second. What is your version of bet hedging against unexpected stress? Do you have a backup plan running in the background, consuming just a little bit of energy, just in case everything goes wrong? Because your gut bacteria certainly do.
It's a good lesson.
Now, we've explored the master breaker, this genome-wide shuffle. But what happens when the bacteria just needs to make a subtle adjustment, not a massive emergency protocol?
That brings us to the site-specific EPVs, the localized dimmer switches. The researchers noted that these site-specific changes are heavily enriched at the promoters, those docking stations for genes we talked about.
Right, the turf war.
This physical location is what proves the competitive binding model. The MTACE and the transcription factor are literally wrestling for control of that one specific promoter.
And they saw this dimmer switch effect happen in real time in the infant gut too, right?
They did, but this time they looked at a different bacteria. Bacteroides ovatus, also under amoxicillin stress, they mapped the methylation and found significantly increased tags at 3 very specific localized areas.
Okay, let's go through those 3 areas. What was the first?
First was near a gene for sigma factor 70. Now, a sigma factor is essentially a cellular factory manager. It tells the bacteria which basic survival processes to prioritize. By methylating its promoter, the bacteria changes its core metabolism to bunker down.
Got it. So adjusting the internal thermostat, what was the second area?
The second was near the SUSI and SUSTI genes.
SUSI and SUSTI.
Right. These genes encode outer membrane proteins. They are essentially the receptors and locks on the outside of the cell. By wrestling for the dimmer switch at this promoter, the bacteria alters its outer surface proteins.
So it changes the locks on its doors so the antibiotic can't recognize it or dock onto it?
Exactly, a microscopic change of clothes. And the third area was near the FeOB gene.
Okay, FeOB, what does that one do?
Well, FeOB normally regulates how the bacteria responds to low phosphate, but its secondary function is controlling biofilm formation.
Ah, biofilm.
Yeah, a biofilm is a thick, slimy, protective bunker that a community of bacteria builds around itself. By tweaking the methylation at FeOB, the bacteria initiates the construction of this physical bunker to ride out the antibiotic wave.
So Bacteroides ovatus uses these tiny, localized dimmer switches to tweak its internal metabolism, change its outer locks, and build a slime bunker, all simultaneously.
It's incredible multitasking.
But the researchers didn't just look at antibiotics, which, is a massive stressor. They also looked at something much more common: fecal microbiota transplants and commercial probiotics.
Right, and this is where the research becomes highly relevant to anyone who takes a daily supplement. They set up an in vivo mouse model using germ-free mice.
Germ-free meaning sterile.
Exactly. These are mice raised in sterile bubbles. They have absolutely zero bacteria in their gastrointestinal tracts. The researchers took a commercially available probiotic called pendulum glucose control, which contains several beneficial live strains, and they fed it to these mice.
Okay, wait, I have to ask a logistical question here. If the mice are entirely germ-free, their guts are basically empty real estate, right?
Empty real estate, yeah.
So when you give them the probiotic, how do the researchers know the bacteria are actually using epigenetic switches to adapt to the gut environment? Couldn't the bacteria just be reacting to the fact that they are no longer freeze-dried in a capsule on a warehouse shelf? How do we know it's a specific defense mechanism?
That is the exact right question to ask, and it comes down to the sheer specificity of the genes being targeted. The researchers tracked the probiotic strains as they moved from the capsule into the living gut. They didn't see a random awakening of all genes. They saw a dramatic, highly specific shift in methylation at the promoter for a gene called KPSS.
Okay, KPSS. What does that one do?
The KPSS gene is the master builder for the bacterial capsule.
Wait, like the pill capsule?
No, the biological capsule. The thick, sugary, outermost layer of the bacterial cell itself that allows it to stick to things in the gut.
Oh, I see. So going back to our musical chairs analogy, while the bacteria are sitting dormant in the probiotic capsule in your pantry, the music is playing, the environment is stable, and the methylation pattern at that KPSS promoter is locked in one state.
Exactly.
But the second you swallow that pill, the bacteria enter the dark, acidic fluid environment of the stomach and intestines. The environment radically changes. The music stops.
And suddenly, a transcription factor beats the MTACE to the chair at that specific KPSS promoter. It blocks the highlighter tag.
And by doing that, it completely alters how the bacteria builds its sticky outer armor, allowing it to successfully attach and engraft into the gut lining.
Exactly. It isn't just waking up from being freeze-dried. It is actively calculating its environment. The site-specific EPVs act as a localized, fine-tuned adaptation mechanism. It's not a panic button like the master breaker. It's a calculated adjustment to local conditions.
Whether that is forming a biofilm to survive a mild chemical stress or tweaking the outer capsule to colonize a brand new host.
It's all about fine-tuning.
So next time you take your morning probiotic, think about the absolute turf war happening in your stomach. I mean, Intense games of molecular musical chairs being played out across millions of cells just so those beneficial bacteria can tweak their armor and survive the journey. It really changes how you view a simple daily supplement.
It really adds an entire layer of complexity to the microbiome that we were completely blind to until this technology existed.
So bringing it all together, what does this actually mean for us? We've seen Akkermansia bet hedge against amoxicillin by completely scrambling its master breaker. And we've seen probiotic bacteria tweak their local dimmer switches to colonize an empty gut. Why is this 2026 taper such a massive paradigm shift for biology?
Because it proves that having a diverse, heterogeneous population within a single bacterial strain is a highly calculated evolutionary strategy. And epigenetic phase variation has massive, undeniable advantages over traditional genetic mutation.
What kind of advantages?
Well, when a bacteria physically mutates its core DNA sequence to become resistant to an antibiotic, there is almost always a mutation cost.
A permanent trade-off.
Yes. It might survive the drug, but because its core engine is now permanently mutated, it becomes permanently slower at digesting food or weaker when competing with other bacteria. But EQV has zero mutation cost because the underlying blueprint never actually changed.
Right, this bye just takes off the jacket.
Exactly. Furthermore, as we saw with the ciprofloxacin twists, it is entirely reversible. When the stress disappears, the bacteria just swap the highlighter pens back to normal. And finally, the genome-wide EPV allows for the coordinated simultaneous activation of dozens of survival genes globally. It's a highly sophisticated defense network.
And the direct medical application of this is incredibly exciting. I mean, if we now have the algorithms to know exactly how these bacteria use epigenetic tags to survive stress, it opens the door to engineering completely customized, stress-resilient probiotics.
Absolutely.
Instead of just swallowing a capsule of generic bacteria and hoping a few survive the stomach acid, we could theoretically manipulate the epigenetic states of those bacteria before we even manufacture the pill.
We could prime them. We could ensure the entire population is locked into the ideal M-phase disguise necessary to survive our specific stomach acid, bypass our immune system, and colonize the gut perfectly. We are moving from a scattergun approach to precision-programmed biology.
Which brings me to a final, slightly mind-bending thought to leave you with today. If our gut bacteria are constantly shifting their epigenetic states based on our local environment, meaning, the specific foods we eat, our daily stress hormones, the tap water we drink, the medications we take, are we accidentally training our microbiomes to become highly resilient superadapters?
It's a fascinating question.
And if we map this completely, could we one day intentionally program the epigenetic states of our own native gut bacteria to cure inflammatory diseases or fix metabolic disorders, all without ever permanently altering their underlying DNA?
I mean, this paper suggests that the control panel is already there. We just finally learned how to read the switches.
The invisible shapeshifters inside us are infinitely more calculated than we ever gave them credit for. Keep questioning. Keep exploring the deep dives of science. And remember, the next time you pop a probiotic or take a prescription, there is an entire epigenetic war room calculating its next move right inside your gut.